probiotic alternative to antibiotics in poultry industry. Okay, the objective of the group here today is to create healthier animals, including ourselves. Not use antibiotics because of the deleterious effects that can happen through that use, plus the resistance factors and all that business. Good diet, good enzymes to help the diet. The idea is you want a healthier animal, help create a healthier microbiome. In a human, 90% of us are microbial. 10% are our somatic cells. 90% is microbiological. Yet we use that. That's a fact. So we create a healthy microbiome through nutrition, proper enzymes and what have you, proper nutrition. We'll have a healthier gut and consequently a healthier animal. About 1985, for reasons I've forgotten, we started talking about probiotics. I was in Sweden at the time. My colleague there had a collection of lactobacilli from healthy pigs, and his job was to determine among this collection, is there something unique among some lactobacilli that are different, that could be good probiotics? And he didn't know how to select for the right probiotics, and I left him with some ideas how to select for something different among these lactobacilli that could account for them to become a probiotic, a legitimate probiotic, to help establish a good microbiome in whatever animal you're interested in. So we got started on that, and the group that, these 6 folks, 3 professors and 3 students, are the core. I want to someday write a magnus opus giving them the credit for this, the discoveries I'm going to talk about. That actually, well, and of course, in this group of poultry scientists that help us do all our experimental work here at the NC State labs. In 1987, we already had the cultures. We had cultures, actually they're pig cultures, but we learned how to screen for lactobacilli from other animals using a little trick, namely the ability of these lactobacilli to make a particular antimicrobial. We used that antimicrobial to be able to identify other organisms that had the same species. In 1987, we asked the question, if oral administration of viable poultry Lactobacillus reuteri, this is using a chicken or turkey reuteri, Lactobacillus reuteri, I'll just call it reuteri from now on, would enhance the health and economic value of chickens and turkeys. Our idea was, here's a turkey. Here's what we hoped our reuteri would do. Here's what we hope our reuteri would do. Here's a good healthy turkey, and a turkey without our Lactobacillus reuteri. Okay, that was our goal in these studies starting in 1987. Safety studies were conducted first. All right, here we have an organism, we don't know anything about it, we know it produces an antimicrobial, so what? Is it safe? So we ran all kinds of safety studies. In fact, we've run, here's chickens and turkeys. Here's the chicken and turkey, but we ran the safety studies using human strains of Lactobacillus reuteri in the States, HIV positive adults, to make sure that our reuteri wouldn't be a pathogen for someone with a compromised immune system. Premature children, children hospitalized, children all over the world, pigs in Peru, chickens, turkeys in US and Europe, and even rodents. If you used host-specific strains, no problems. They were safe as could be. Okay. Yep. Okay, safety is one thing. Efficacy. Would this probiotic, to help the microbiome develop normally and healthily, efficacy studies have been the down road of probiotic studies. Probiotics primarily used in animal industries, cows, pigs, chickens, turkeys, and so forth. Most of them aren't very successful. They cannot obtain data that's statistically significant to say, aha, this is working. Sometimes your probiotic works, sometimes it doesn't. That's been a whole history of probiotics since Metchikoff developed a concept in 1916 or whenever he did. this concept. And this has been a plague on the industry, no statistical data. So we decided, okay, if this roid is going to do something, we have to establish efficacy to provide statistically sound scientific evidence that the microbiome can be manipulated. And this is what we want to do. Get a healthy microbiome, you have a healthy animal. To enhance establish a microbiome can be manipulated to enhance human and animal health by probiotic interventions with viable cultures of Lactobacillus reuteri. That was our specific objective. And we were working at that time with chickens and turkeys here at NC State. Okay, how are we going to determine efficacy? How do you do that with statistical significance? What we did, we fed off of Koch's postulates, which most of you know. Koch, how do you define a pathogen? How do you identify a pathogen? His postulate said, find that microbe present in a disease host, isolate a pure culture of that microbe, administer it to a healthy host, induce the same disease, and then re-isolate the same microbe. You have Koch's postulates. That organism causes that disease. Okay, our question was, what organism now can help the animal as a probiotic, as a positive to its microbiome? So what we did now, the Dobrogosz Casas postulates, and I have to say a sad note here, are Ivan is a fellow Penn Stater friend of mine from way back. He died 10 years ago. This was a wonderful microbiologist, a wonderful human being, and a wonderful businessman. Helped us get moving out of the probiotics into the actual industry, in the turkey industry, poultry industry, and so forth. But at any rate, he and I developed this reverse of Koch's postulates. We said, use a microbe present in a healthy host. That makes sense. Isolate pure culture of that microbe, which we did. Knew how to do it. Took a lot of finagling to figure out how to isolate specific Lactobacillus reuteri from these animals. We could do that. Administer it to a disease-challenged host to prevent the disease with statistical significance. Re-isolate and show that same microbe in greater numbers in treated host than untreated host. It's a reverse of, but it's a beautiful way. If we could establish that with statistical significance, we have then established a true probiotic, not just a probiotic. In mid-1950s, 1980s, probiotics were considered frauds, snake oil, on and on and on. People were using it. Farmers were using probiotics, but nobody was happy with them. There was no statistical evidence that they were working. Okay, we decided we're going to work with statistical evidence. And here's just, I'll go through very quickly some slides. I don't want Jacob to stand up here. I'm going to go through. This is some data. Here's effective Lactobacillus reuteri. This is chicken reuteri or turkey reuteri, whichever it is. Mortality on Salmonella typhimurium-induced mortality in poults. Here's the age, zero age with salmonella. With salmonella, we see the deaths about 40%. With reuteri, fed reuteri at the same time per os, we can see a tremendous effect on mortality. Here's where you look at the gut of those animals. And here, this is a big surprise. This histology was done by another professor at NC State, blind studies. And she found something strange here. Here's a healthy chick. Here's a healthy chick. It's ileal type tissues. Bigger ileal, bigger crypts. That's usually a sign something's wrong. There's an infection going on. The animals are being stirred up for infection. No, these are perfectly healthy animals. But there's more surface now in the gut for absorption of nutrients and so forth. So it's interesting, but something behind that. Why the ilia bigger? This is another sort of a similar experiment this was with weight gain and body weight gain, livability. Again, Salmonella typhimurium and cold stressed poults. Cold stress pulse, mortality is bad, weight gain is low compared to the roid-treated pulse. Things are working out. This is just showing competitive exclusion. When you add roid and salmonella to these animals at birth, here's with roid present, the salmonella counts go down. If you inoculate after 5 days or so, the same competitive exclusion. Roid is warding off the salmonella. By 7 days or however that is, it doesn't make one. We see competitive exclusion. Roid is antagonizing the population of salmonella in these experiments. Here, now we're getting some antibiotic material. Gentamicin was suggested we try gentamicin. Here now is a histology of salmonella typhimurium infected ileum tissue. We see some inflammatory damage going on. Here's now the same tissue with gentamicin. Antibiotics work in this particular case for this particular challenge. Here's salmonella typhimurium and roid. So we see the roid now doing what the antibiotic is doing. Substitution we can substitute for the antibiotic. And this is now adding them all together, good healthy animal. The inflammation is compromised with roid or antibiotics. So the question is, can we forget about the antibiotics and just use the roid? That was our ultimate goal, and we'll see what happened there. This is just the effect of lactobacillus roid on turkey body weight distribution profile at the time of transfer to range houses. We're getting into the commercial aspects now of is probiotic worth it? The growers, at the time that the birds are being transferred from whatever stage they're at to the final stage of growth, they'd like the animals they'd like to be evidence that roid actually did make a healthier animal, mortality-wise and body weight-wise. And that's exactly what happened. At that time, we can see that you have larger animals at that time for transfer to the other houses. Here's sort of a summary that we presented to the growers of data. I think it's effective roid and gentamicin on death and morbidity of newly hatched broiler chicks challenged with enteropathogenic E. coli. Oh, this is just another similar experiment. Absolute controls, good body weight, low mortality. Each E. coli challenged, body weight drops, almost 10% mortality. Gentamicin, that helped, less mortality. Here's roid, healthier, bigger birds, and better than gentamicin in terms of mortality. Gentamicin and roid, bigger birds, and no mortalities. Two working together, but roid by itself is doing a pretty good job. This is a summary of roid treatments on performance of commercial turkeys done in 1998. This kind of data the growers loved. Flock mortality, this p-values, everything statistically significant. Improvement, 2.8%. Body weight, 2% better. Feed conversion, they love this number, 9 points. That means something to the growers. Didn't mean much to me, but they were happy with that kind of a number. Grade A, 9% increase in grade A, whatever that is. But these are evaluations by the growers. Things are looking very good for us. Okay, what underlines the mode of action? I'm going to go through this very quickly. This is another study we made that was surprising. It's really led to the mode of action discovered by us and other people later on. We took, these are now roid stimulate, we found that roid stimulate CD4 cells production in ileum of healthy and typhimurium infected chicks. Okay, here's CD8 cells. We're counting the cells. And here with stress-brooded control, stress-brooded roid normal brooding with roid CD8s didn't change. However, when we looked at the CD4 cells, normal brooding controls, here's a CD8 count in tissue. Here's now with roid stimulating the CD4 cells. In stress-brooded animals, same thing. Roid is doing something to the healthy animal or the stressed animal. Roid is affecting the physiology, the immunology of the animal. Actually, this was done in Europe many years later in humans. they used labeled CD4s and found that the human ileum is a repository for the human rotori. Human rotori is actually mother's milk rotori. Mother's milk rotori is isolated from mother's milk, approving mother's milk. The patients are the experimental patients in this case were given labeled rotori, and then these studies were done. And where did the rotori end up? Pretty much in the ileal areas, the largest part of the small intestinal system. Similar to sound chickens, pretty much the same thing happened now in humans, which of course we're now in the human business. I'll show you why we didn't get out of that business, the chicken business. This is let me forget this one. No, let me just mention quickly. This is a gut, any gut. Here's the microorganism in the gut, in the lumen of the gut. If the right organisms are there and they interact with certain toll receptor cells, here, dendritic cells, that have certain receptors on them, that interaction with the organisms with those dendritic cells creates a good, healthy state. If there's a genetic problem with the toll-like receptors, no matter if the good organisms are there or not, nothing's going to happen. And if the organisms aren't there and you have good receptors, nothing's going to happen. So adding a probiotic, for example, is what we're doing here, or we add a probiotic to this, would convert it to this. That's the theory, sort of immunological theory behind that. Years later, different labs were grabbing on the rotori, looking at mode of action. They knew immunology was involved. Here's one of the summaries of dendritic cell signaling influences production of regulatory T cells. So when a whole new member of the CD4s come in, regulatory T cells. And the role of the regulatory T cells, here's a pathogen attached to a dendritic cell, which is sticking right in the lumen of an intestinal tissue. It's reading what's inside the gut. It's reading the organisms, it's reading toxins, it's reading everything that it has binding sites for. So here's a pathogen attached to a dendritic cell. That tells that dendritic cell to stimulate the production of cytotoxic cells, cytotoxic cells, effector cells that now attack the inflammation. Pathogen's there, you need information to get rid of the pathogen. However, if there's lactose rotori present, that actually stimulates the production of IL-10, which is an anti-inflammatory cytokine. That shifts, here's now the rotori and the pathogen, both attached to the dendritic cell. Now, with rotori present, that dendritic cell is starting to make T regulatory cells, with turning on the FOXP gene, and now you have T regulatory cells. So now you have a balanced effect. Here you have a cell trying to get rid of the infection. Here the cell's trying to prevent an immunopathology. When infection goes wild or the infection can't be controlled. A nice balance. That's what this particular probiotic is doing. There's just another view of the same thing with a little more detail. My mouth is dry. You're standing up already. All right. Just another minute. This is just look at the genetic level. These are dendritic cells. Here's the pathogen. Pathogen turns on a series of events that actually result in the formation of nuclear factor kappa beta. That translocates itself into the genome, and it's a pro-inflammatory process. Rotori now sit in that same gut with a pathogen attached to the dendritic cells, and it prevents this NF-kappa beta from translocating across the cell, the chromosomal membrane, the nuclear membrane, and therefore you don't have the inflammatory reaction. There's the balance, the T-cell regulatory balance instigated by this particular probiotic. And I'm going to just, we know that rotori makes a lot of, the antimicrobial effect is an antiviral effect. It's antibacterial, antifungal, antiprotozoal, anti-inflammatory. These are sort of extra things. An arsenal, rotori has a larger arsenal than just being anti-inflammatory. Rotori produces various prebiotics, which also can be used as probiotics, prebiotic picture. It produces vitamin B12, production of CLA, conjugated linoleic acid, been implicated in mammary cancer in some mouse experiments. Production of roiderin, that's the compound that we discovered and have patented, but also a German discovered to make some other antimicrobial proteins as well. Produces gut adhesive protein so that roiderin has an attraction to certain sites on the surface of the organism. Production of nitrate reductase, nitric oxide, how important that is in terms of hormonal repercussions. Roiderin inhabitation may counteract carcinogenic processes involved in epigenetic control of resident T-reg populations. And it's 2014, cancer regulation is coming into the picture. With T-regs, which is a simple little organism, is influencing in the microbiome. And I think I'm going to quit right here, just one moment. The Dobrogosz family, this is our family about 10 years ago, maybe 20 years ago. There's me and there's my lovely wife, the only one with the hat on. Why'd you wear a hat that day? Oh. This young lady here is now going to be a senior in high school. So this was quite a while ago. Anyway, that's the Dobrogosz family. We use roiderin. Most of us use roiderin. I've got a daughter-in-law who needs roiderin because she was treated for Helicobacter pylori, stomach infection, treated with antibiotics, reduces the Helicobacter pylori infection. To keep that Helicobacter pylori population at bay, she takes roiderin, because roiderin and helico have the same binding sites in the stomach and duodenum. So there's an interesting factor there. Okay, that's we use roiderin. So then, question, why is lactobacillus roiderii probiotics good for the Dobrogosz family, but not accepted by the poultry industry? The poultry industry, no matter what we did to the data, no matter how cheap we made the product, we couldn't go cheap enough. We had to go down below the penny level before they even talked to us. So that's my frustration. But now we're back, maybe we're back looking at poultry again, hopefully. But we're working with humans now. It's working with humans, and hell with poultry at this moment until we can get the cooperation from the poultry growers. We can get rid of antibiotics, hopefully, with research like this, and at the same time get better animals, as long as they also get the enzymes from Jason's company. Thank you. Sorry I took long.